Hello. Today on Learning Image Analysis, we'll be revisiting our topic, highlighting desired features in your image. So the image that we have today is one of colonies of cells in a petri dish. And we want to get a count of the number of cells that are present, as well as the size of the colonies, and also the confluency. The confluency is the percent coverage of the entire field with colony. So, our learning objectives, first we'll briefly touch on proper image illumination. We can see in this image there's overexposure in the center, underexposure in the back, and what might be the cause of that. And then we'll, as we discuss filters that we can use to compensate for this improper image illumination, we'll talk again of being aware of filter side effects. And then we'll get into combining multiple segmentations from the output of multiple filters. And lastly, a little bit of extra, we'll be using segmentations as masks for subsequent processing. Once we find individual colonies, we'll use those colonies as masks for finding all the individual cells within the colonies. So first, let's talk about the illumination of this image. We can see that there's overexposure here in the middle. A lot of the boundaries between cells aren't properly defined. And then in the upper left and right corners, there's shadowing. And this causes a number of issues for the segmentation. We're not going to be able to get an accurate count of the number of cells because we simply can't accurately determine where the cells are, at least not in an automated way. And then we'll have to do some adjustments to make sure that we get a reasonable segmentation here in the bright portions and in the dim portions. And we also have this haloing that's going on around our colonies. And these are going to cause some issues for us as well. Now in this particular image, a lot of these symptoms are very classic to what's known as improper kolar illumination. So in bright field microscopy, kolar illumination means this set of lenses here in the light path, set of lenses and diaphragms that perfectly defocus the light when it's hitting the sample. That way you get nice, even illumination. And what we see is that this field diaphragm and the collector lenses are not set properly. If we bring our image back up here, this shading that we're seeing here is actually the edge of the field diaphragm. Because this collector isn't centered properly, these edges are intruding in the upper corner. Also, you see that it's fuzzy rather than the sharp, true edge of the diaphragm because the collector is out of focus. It's not where it's supposed to be. So, to really fix this image, you need to focus the diaphragm and focus this whole collector by using the edge of the diaphragm, get it nice and sharp, get it centered, and then expand. That way you can get even illumination across the image and it'll produce something with much less artifacts in it. So it's very important to know how to properly illuminate your sample and how to properly use the instrument that you're using. Now that aside, let's move on to actually segmenting this image. And so what we have to do is try to get these cells and these cell boundaries identified without getting them confused with this halo around the outside. And this is going to be difficult based on the quality of the image, we're not going to be able to get it perfect, but we'll do the best we can. So our plan of attack will be first to try to pick up these edges, these outlines here, using the dark texture that we talked about before. So that'll pick up these edges, but it's also going to pick up some of these halos. So what we'll do to compensate for the fact that dark texture is going to pick up some of these halos is, in a second step, use the bright texture function. The bright texture is going to pick up the individual cell bodies, but it might also pick up some of the space between the halos, because this would also be found as a bright piece of texture. However, if we combine the two, we'll see that we can get the cells identified, because the cells will be found pretty evenly in both the bright texture and the dark texture. But a lot of the halos will be found only in the dark texture, and the space between only in the bright texture. So if we make an intersection between the two of them, we'll then end up with just the cells themselves. Of course, it's not going to be perfect. As I said, there's only really so much we can do with this image, but it'll be a lot better than using just one or the other. So if we start here, we'll use our bright texture function. Now, if you're not familiar with it, what it does is it finds regions of the picture that are brighter than the surrounding pixels, but these regions have to be smaller than a certain window size. So if I set my window size for 3, we only find pieces that are about 3 pixels across. And of course, it's a little bit fuzzy with that boundary. This number isn't going to exactly correlate, especially because our windows are square. But as we increase this window size, we'll find larger and larger items. And so when we get to 9, then we start seeing that we've picked up a lot of the cells. And really, we're comparing it to how much of the back of this space between the halo and the cells that we pick up. So above 9, we start to get some extra stuff over here, and we start to really lose a lot of improvement on the cells themselves. So really, 9 is pretty good for our purposes. Okay. So now we'll take this processed image and do a basic threshold to select all the bright cells. And of course, we have a piece here where this space between the cells and the halo was found. We knew that was going to happen. But we'll do some just standard cleanup here. If we reject first tiny features to get rid of some of the stuff around the outside. And let me also point out before I forget, another thing that the bright features, the bright texture finds, is concave edges. So we see here this concave edge, which isn't a cell, but it's between 2 dark pieces. So this also gets found as a piece of bright texture. And this is, of course, will be something that the dark texture will not find. So it'll get, again, evened out when we combine the 2 different methods. So we have our cells here roughly selected. If we fill the colonies in with a dilation here, we'll dilate by 5 to fill in all the holes in the middle. Then we'll erode by 1 or 2 just to get our selection down to around the edges of the colonies as close as we can make it. Okay, something like that. Now we'll save this away for later and go back to our original image, and now we'll use our dark texture to find our cells with our second method. So if we dark texture, and again, the window size determines the feature size that is picked up. So the edges of these cells are, of course, smaller than the cells themselves, and with a window size of 5, we highlight them pretty well. If we go up a little bit larger, we see this halo, which happens to be a little bit larger, start to show up. So if we bring our window size down, 5 seems to be a pretty good number for finding these edges. And again, we'll do another basic threshold here to pick up our cells. All right, now let's do some cleanup. Again, we want to try to fill all this in. So we dilate by, let's start with 5 again, the same as the previous. That looks pretty good. It's filled in at least the edges of all of our colonies. The more we dilate, the more we're going to end up joining individual cells, joining things that aren't supposed to be touching. So really, we just want to go far enough that these colonies are whole individual units, because then we can fill in these tiny little holes later. We don't need to have them completely filled in just with the dilate function. Right? So 5 seems to be the minimum number that will accomplish what we want without doing too much extra connections. So now if we fill in all these holes and do an erosion. Three, 4, 5, 6, about there, so we're at the edge of the colonies. So now what we see, if we just do a quick scan, we notice that in these really bright portions, there's some cells which aren't being selected. Okay, this edge of this colony here is being missed. And the same is true here. Now this is because the overexposure is so great here in the bottom right. But we'll see if we can do a little bit more selection in order to get this a little better without starting to over-select too much of the halos. So if we take our basic threshold and taking care to look at these, right, while also watching this halo portion here. Okay. Comparing these two, maybe 9 would be a better number than the 12 that was auto-selected. And now when we look at our result, we have a little bit better selection of these cells. Again, not perfect, but better. And we have a little bit of extra dark texture here on the edge. This little piece that was gotten, which shouldn't have. But this is something that'll get knocked off when we intersect with our previous bright texture. So now we have our 2 segmentations, and this is stored in our companion. The bright texture is, And if we now intersect that with the result of our dark texture, we will have our result. And we see dark pieces like this get cut off, and some of this concave surface here, which was found with a bright texture, gets cut off because the dark texture didn't find it. So it's not perfect. Of course, we do have little pieces here and there, but for the quality of the image, it's pretty good, about as good as I think we're going to get. So now let's do a little bit of cleanup. We'll reject some tiny features that were produced by this intersection. Do a reject features of 5 and accept. And then whenever you do an intersection like that, you get some sharp edges on the edge. And so we'll remove these sharp edges with our smart erosion. So any pixels that are surrounded by less than 5 selected pixels will be removed. So anything that's a sharp edge that has at least 3 or 4 empty pixels around it will get eroded away. And it's a small thing, but it's easy to do, and it will help the segmentation later. So now we have our colonies. And we can perform measurements on these colonies. We can measure the area fraction. That'll give us a confluency measurement. And we can also measure the the size of these colonies. Now, of course, we don't want to measure the size of colonies that touch the edge because they're not being seen. So if we reject those edge features and we have just complete colonies, we can get a size measurement of those. So if we do this as a feature measurement, excuse me, let me set it as a layer, complete colonies. And we'll measure the area of each colony. Now, I mentioned the next step was to find the individual cells within the colonies. And we're going to use our colony selection as a mask for that process. So let me set this colony away in memory and call our original image. And now we're going to use an adaptive threshold to find these individual cells. They will be brighter than the surrounding pixels. And we're using adaptive rather than the basic threshold because of this intensity difference across the image. If you remember, that's one of the tricks that we learned in a previous lesson. So we have our adaptive threshold, and we only want to do selections that are within the colonies. We don't want to select stuff around the outside, even though they are pixels brighter than the surrounding pixels. So we're going to use our companion image here, which is our colonies, to act as a mask. So we'll only consider pixels that are within that mask. So if we set our percentage here, say at 100, we want bright pixels. Okay. And you can adjust this number a little bit. We want to have our colonies be complete enough here in the center, our individual cells, but we don't want to over select so that it's hard to cut these cells apart later to get a count of them. So really about 100.5 is a good balance between getting complete cells and still being able to separate the cells from each other. Okay. And now we'll do our cleanup. We'll run our separate features step. Okay. And that will cut these colonies apart. Excuse me. Cut these cells apart into individuals. And then we'll do a reject features to remove some of the small stuff on the edge. And lastly, we'll clean up some of these sharp points, again with our smart erosion. And these will be our cells. And now we can measure the count of these cells. Here, let's select our layers here. All right, for area fraction, we want colonies. And for count, we want cells. For those feature measurements, we want just complete colonies. So now we have our result. We have, say, about 25% of the image filled with colonies, and the complete colonies here have an average area of 2,410 pixels, and we have roughly 1,500 cells across the entire image. Now, the takeaways that I want you to have from this lesson: one, of course, you want to make your original image as good as possible. Be familiar with your instrument, be familiar with how to use it optimally so that you can get the best results. It'll save you a lot of headaches later. And then, when you're using some sort of filter to act on an image, make sure you know the side effects that filter will have and how those side effects will show up with your particular image or set of images. Because once you know, once you're aware, then you can do things to compensate. You can use a different filter with different side effects and take the intersection between them. Or you can do some special cleanup to remove those side effects if you know that they happen to have a particular quality about them that you can target with something else. Some other type of filter, some other type of segmentation. So that's it for today. Next week, we'll talk about removing distractions from your image. I'll see you then.